Yes, in the previous video I have explained you about the classification of reversible inhibitors like competitive, non-competitive and uncompetitive. And the irreversible inhibitor is also called as suicide inhibitor, and we will see one by one. So coming to the competitive inhibitor, it just compete with the substrate. The competition is held between the inhibitor and substrate. So the inhibitor competes with the substrate for the active site in the enzyme. So the chemicals which resembles the enzyme normal substrate structure. So it looks as like the substrate and it compete with the substrate for binding at the active site of the enzyme. So an enzyme having a small part of the region is called active site, where substrate will bind and it just forms the enzyme substrate complex and product. The inhibitor looks like the substrate, it just competes with substrate for the binding active site of the enzyme. So any compound that bears the structural resemblance of the normal substrate, it is called competitive inhibitor. So this inhibitor is not acted upon by the enzyme, but it just prevents the substrate from binding into the active site. So this is the definition of the competitive inhibitor. And you could see the normal enzyme reaction is the enzyme, the substrate will bind at the active site, enzyme substrate complex formed, and enzyme releases the products, and the free enzyme waits for the another substrate for the activity to be continued. In inhibition, you could notice the inhibitor having the same shape structural resemblance of the substrate. In the substrate place, the inhibitor will bind and no longer the substrate is bind to the enzyme, hence no product will be released there. So the enzyme binds with the inhibitor and inhibitor competes with the substrate, and the original normal substrate will can't able to bind into the enzyme, hence no product get released. So a very good example for competitive inhibitor is the malonate. So malonate, it is the competitive inhibitor where it is having the structural resemblance of the substrate succinate. So you could see the structural formula for succinate and malonate here. Succinate having COOH, CH2, CH2, COOH, where malonate having COOH, CH2, COOH. Only the CH2 will vary from succinate to malonate. So here the malonate will bind to the succinate dehydrogenase enzyme in the Krebs cycle reaction. So succinate converted into fumarate with the help of succinate dehydrogenase enzyme. So in succinate place, malonate go and bind, so that malonate does not form the fumarate, hence no product is there. So malonate acts as the competitive inhibitor because it is having the similar structure to succinate, and this inhibitor malonate binds to the active site of the succinate dehydrogenase enzyme and inhibits the reaction, hence no fumarate product will be formed there. So this is the structure. Enzyme succinate gives succinate enzyme complex and the fumarate plus enzyme get released. Where succinate place, malonate will be binding, enzyme malonate complex, no products formed in that reaction. So this is a very good example for competitive inhibitor. Next, coming to the non-competitive inhibitor. So non-competitive inhibitor attaches to the allosteric site, which is not an active site, because it is any site on the enzyme but not the active site. So it do not enter the active site but binds to the another part of the enzyme, causing the enzyme and active site to change the shape. So this type of inhibitor will bind to the another part of the enzyme, not the active site, and when binds it changes or alters the shape of both the enzyme and active site, so that no substrate will no longer fit into the active site properly, and it also result in the three-dimensional structural alteration. So here you could see the part B inhibition. So active site is there, allosteric site is there. Allosteric site is the any part of the enzyme but not the active site, and the blue inhibitor will come and bind in that allosteric site. It causes changes in the active site. You could notice the active site shape, which is in the V form, and after binding of the inhibitor it just forms U instead of V. So the substrate will no longer, because the substrate having the different shape and the enzyme active site having the different shape, and the enzyme no longer will go and bind. So this is the example for non-competitive inhibition. And non-competitive example, it is cyanide, it is the non-competitive inhibitor. This cyanide mainly binds to the enzyme of cytochrome oxidase, and it is mainly for the production of ATP. So for production of energy during cellular respiration, this enzyme is important, and this cyanide is the inhibitor which mainly binds to this cytochrome oxidase enzyme, and it just prevents the electron transport chain and oxygen, and it stops the electrons from being passed to the oxygen. So without ATP energy, cells cannot perform normal respiration, and it finally leads to the hypoxic anoxia. Hypoxic anoxia in the sense low oxygen environment, where our body receives low oxygen or completely absence of oxygen, which further finally leads to death condition.